I'm Vandana and I'm a postdoc at Massachusetts Institute of Technology in the group of Dr. Bevan Engelward. Our genome is constantly subjected to DNA damage. Can you imagine that to combat these DNA damaging episodes, we can have a trillion DNA repair events happening every second if we look at our own body? We can admire the fact that these DNA repair events helps us to stay healthy and our DNA free from cancer as one of the many benefits of the DNA repair machinery. Can you take a moment to think what happens if these DNA repair events go slow? Will this lead to excessive DNA damage or slow DNA repair? Is it possible that people who have a problem in DNA repairing proteins are more vulnerable to environmental toxins found everywhere around us, in the air we breathe, in the soil from where we get the food, and the water we drink? For example, a chemical called NDMA was found in Wilmington, Massachusetts, where incidence of cancer were above average. Here at MIT, I am working on many such important questions related to environmental hazardous chemicals and their effect on us. I closely study the effect of genetic alterations and environmental risk factors using a novel in-house comet chip platform that Dr. Bevan Engelward discovered. This small stamp that you see in my hand can test at least 96 different samples in a very short span of time. You see, this stamp has hundreds of protruding peaks. It is not visible here, but under the magnifying glass, you can see it. I pour a semi-molten gel in a system like this and put the peck-containing stamp on top of it. When the gel solidifies, the stamp is carefully removed and we get a chip with microfabricated holes in it. Then I load DNA-containing samples onto the chip that can be damaged due to different conditions. And we put this chip in between two oppositely charged electrodes. The DNA moves into the direction opposite to its own charge and we get comet-like shape. Many DNA repair proteins help in repairing our DNA. To study how genetics can change the effect of environmental toxins, I am creating biological models. I am using radical CRISPR-Cas9 technology to create problems in some very important DNA repair proteins like XRCC1 and Pol beta. I want to see if problematic DNA repair makes cells more susceptible towards environmental hazardous chemicals, for example, NDMA. After the end of the study, I will be able to determine how problems in the DNA repair proteins change our cells' response when they interact with environmental toxins using comet chip platform.